Welcome back. In today's presentation, I am going to explain Mendel's laws of inheritance. Video 1, chapter Principles of Inheritance, the 5th chapter. This is Gregor Johann Mendel. He was an Austrian monk born in 1822 to peasant parents in Austria. He was very poor. He joined the monastery and he was serving as a teacher, and during his free time he used to conduct experiments on garden pea in the kitchen courtyard, and nearly for 7 years he was conducting and experimenting on this garden pea, and he came out with the laws of inheritance. He died in 1864, but his laws were rediscovered, though he was ignored for a long time. In 19th century his works were rediscovered, and because he was the first to conduct experiments in genetics, he is considered as the father of genetics. Okay. His rules, especially the first 2 laws, he has come out with 3 laws: law of dominance, law of segregation, and law of independent assortment. The law of dominance and law of segregation is the most universal law. It is applicable to most of the inheritance, but some have been deviated. Some of the patterns are deviating from these laws, but because he was the first to conduct experiments in this field in genetics, he is considered as a father of genetics. Okay. Now, the experiment he conducted was on garden pea, Pisum sativum. And the speciality of Pisum sativum is that it had 7 contrasting characters, namely seed shape. The 2 contrasting characters are round, wrinkled. Then seed color, yellow, green. Flower color, violet, white. Pod shape, full, or in some books or in the examination they can give it as inflated, and the other option constricted. Pod color, green, the other option yellow. Flower position, axial or terminal, and stem height, tall, dwarf. So, there were 7 contrasting traits. Now, the first column indicates the dominant traits, and the second column indicates the recessive trait. So, when you learn, also learn it in this order, not in the order of the characteristics, but remember, first you mention the dominant trait and then mention the recessive trait, because this will help us in assigning the alphabets. There are rules to assign alphabets. For example, we are going to work a cross between tall plants with dwarf plants. Now, how should we apply it? So, I told you it is a cross between tall plant with dwarf plant. Now, we know tall is dominant over dwarf. See, stem height, tall is dominant over dwarf. So, we are assigning based on the dominant trait. The dominant trait is tall. Tall, the starting letter is T. So, we are assigning the genotypes with T. So, tall will be capital T, and dwarf will be small t, small t. Now, remember, there are 2 types of talls. Either this capital T is a homozygous tall, or the other option is capital T, small t. This is a heterozygous tall. So, once again, when you are assigning the alphabets in genetics, it is based on the dominant trait, the alphabet of the dominant trait. And remember, the dominant trait exists in 2 forms, homozygous and heterozygous. Homozygous, this is homozygous tall, and this one is heterozygous tall. So, there are 2 types of talls, but dwarf, only one option for dwarf, small t, small t. Now, another example, I am going to assign based on another trait, violet cross white. So, plants with violet flowers were crossed with white flowers. So, now how are you going to assign the alphabets, or how are you going to assign the genotype? Now, which is dominant, violet or white? Violet is dominant. We learnt in the table, violet is dominant. So, you assign based on V. So, this will be capital V, capital V. There is 2 types of violet, homozygous violet or heterozygous violet. And white is only one option, small v, small v. So you understood how to assign the alphabets to get the genotype. So this is the genotype. And what is the physical appearance is indicating the phenotype. So genotype is the genetic constitution. It is represented in alphabets. So this is representing the genotype, capital V, capital V, capital V, small v, small v, small v. All these are representing genotype. Then what is phenotype? Phenotype, as the name suggests, physical appearance. Violet is a phenotype. White is a phenotype. So phenotype is the observable trait, whereas genotype is the genetic constitution, or we can say the alphabets it's indicating. Okay. And always remember, when the dominant trait, there are two types, homozygous and heterozygous. Homozygous where the two factors, Mendel called it as factors, but now it is called as alleles. The two factors are similar, whereas in heterozygous the two factors or alleles are dissimilar. So now you know how to assign dominant the alphabets or genotypes of all the 7 characters. So always remember, represent the dominant trait first and then the recessive trait. And dominant trait, there are two options, homozygous and heterozygous, whereas recessive, it is double recessive. Both the alphabets should be in the small letter. Okay, so this is the rules of assigning the genotype. Now again, getting back to the rules of working a problem. The format of working a problem, any genetic problem will have the following format. First, P1. P1 stands for first parents. Second step, gametes. Third step, first generation, F1, first generation or filial 1. Latin, it is filial generation. Then again, if you are proceeding, if you have to get the F2 generation, again continue, P2. Gametes, second generation. So for your NCERT +2 syllabus, only you are having up till F2 generation, need not go to F3. Now P1, you have to represent the both phenotype and the genotype of the parents. For example, I am going with tall, this is the phenotype and this indicates cross, X indicates cross with dwarf. Now what is represented is the phenotype. Now I have to also represent the genotype, genotype in bracket, which tall. The question paper will reveal the tall, or you have to do trial and error method. So I am going with the option homozygous tall, and dwarf, there is only one option, small t, small t. Next we are going to find the gametes. The rule for finding the gametes is these two alleles should separate. Now, and gametes should be represented in a circle. So here, how many gametes I draw? See, if I am drawing many circles, the gamete will comprise only one capital T. So there is no point of drawing many circles. So only one type of gamete. There may be hundreds of gametes, but there is only one type, that is capital T. Here, small t. Okay. Next step, how to form the generation? Just join these two. This fuses with this. This gamete fuses with small t to form capital T, small t. Remember, don't put it in bracket, don't put it in circle. So we got the genotype. Now what is a phenotype? That when I explain the law of dominance, I will explain what will be the phenotype. Now I want to continue to second generation. Now you will be wondering how to go to the second generation. The question paper has not revealed the second parent. So the rule is this should be selfed. Take this F1. This is the F1 offspring and self it. Capital T cross capital T small t. And I told you I should also represent the phenotype. Phenotype will be revealed only after I explain the rule of law of dominance. Now the gametes. Now the gametes, see, this capital T separates and this small t separates. This capital T also separates and this small t also separates, and this explains one rule on the second law of dominance, second law of inheritance. Okay. So here capital T, here small t, here capital T, here small t. So here we see that the gametes receive only one of the allele or one of the factors. So now we are having two gametes of this parent, two types of gametes and two types of gametes of that parent. Now the cross. This T will cross with both of those T's. Okay. and this small t will cross with these two t's. Okay, so now what are the genotypes? I'm getting capital T, capital T, capital T, small t, capital T, small t, small t, small t. Now only genotypes are revealed. Only when I explain the law of dominance we can understand the phenotype. So as of now I only explained the rules for finding the genotype and the rules for making the problem, for working out the problem up till F1 generation or F2 generation. So once again, the first rule, how to assign the genotype. How we are assigning the genotype? Based on the alphabet of the dominant trait. The dominant trait is axial. We know axial, terminal. If it's given, you must always remember axial is dominant over terminal. That by heart you must learn that box, which are dominant. Most of the traits you can understand is dominant. Okay. So for example, axial, terminal. So we are taking axial is the dominant trait. So we are assigning with the alphabet A. So it will be capital A, and the other side will be small a, small a. But when you are working a regular problem, you are going with homozygous. But if in the question paper they ask, if they say another, there are two types of axial. Remember, dominant trait there exist in two forms. That time you must do trial and error method. Anyway, I am going with the regular model and taking these traits. So you know how to, first rule, how to assign the alphabet. So how to assign the genotypes of the parents. So how it is assigned? Based on the alphabet of the dominant trait, and capital letters and small letters are used. Second rule is how to make the problem. The steps P1, gametes, F1. That's first generation is over. Then to proceed to the second generation, P2, gametes, F2. Remember, F1, when we reach F1, it's over. Now to proceed to F2, what will be P2? Self the F1 parents, you will get P2. Okay, and always remember gametes should be in circle. And second, the last thing you have to remember, the P1 or parents, wherever you indicate genotype, it's mandatory you have to indicate the phenotype. Now I revealed the genotype and phenotype here. See, the genotype is revealed and the phenotype. Here also the genotype as well as the phenotype is revealed. Everywhere it should be revealed, but in the following steps I have not revealed, because I have to apply the law and make you understand how to understand or how to reveal or disclose the phenotype from the genotype. So this is the, I am introducing you to the rules of working out problems in genetics. So he worked on garden P, and his experiments are now called hybridization experiments, and he came out with the 3 laws. Once again reiterating the 3 laws, just the headings: law of dominance, law of segregation, and last law, law of independent assortment. Now we will analyze each law. First law, law of dominance. It has 3 postulates, and I have taken the same wordings of your text because we cannot make it in our own words. Laws and theory should be given exactly as given by the scientist. So the laws has 3 postulates. First, the characters are controlled by discrete units called factors. Okay, any character is controlled by discrete units called factors. Second, factors occur in pairs. And third postulate, in a dissimilar pair of factors, one member of the pair dominates while the other member is recessive. So the third one, in a dissimilar pair of factors, one dominates and the other is recessive. Now I am going to illustrate this. Taking the same example as Mendel took, tall, cross, dwarf, and following the rules of a genetic problem. First is the P1. Now what is revealed? The phenotype. And remember, for a regular problem you go for homozygous condition. So this will be capital T, and this will be small t, small t. Right? Next, gametes. Here the gametes capital T, and here the gamete will be small t. Then F1 generation, capital T, small t. Now I am going to apply the law. We got the F1 generation, all the offsprings formed are heterozygotes, that is having capital T, small t. Now the law states that when 2 dissimilar alleles, see there are 2 dissimilar alleles, capital T is one allele and small T is another allele. Are they similar? No, they are dissimilar allele. When two dissimilar alleles occur in a pair, so now they have occurred in a pair, what happens? One allele expresses itself because it's dominant. So, which is going to express? Capital T is dominant. Capital T is dominant and it expresses itself, while small t is recessive and it is suppressed. So, when two dissimilar alleles occur in a pair, one allele dominates while the other allele is suppressed. So, here capital T dominates and small t is suppressed. So, now can you guess what will be the phenotype of this offspring? Is it tall or dwarf? Tall. So, the phenotype is, it is a tall offspring. But what tall? Heterozygous tall. Clear? So, I have applied the law of dominance. So, to illustrate the first law, you have to work out this cross. Now, what the speciality of this cross is that it is a monohybrid cross. What is the meaning of a monohybrid cross? See the parents, look at the parents, they are differing from each other in one character. So, in how many characters the parents are differing? In one character. One parent is tall, the other parent is dwarf. So, we are taking a cross where their parents are differing in one character. Such a cross is called monohybrid cross. So, another question that can be asked from the boards is, this first law of dominance was derived from which cross? From a monohybrid cross. So, I have illustrated the first law. Clear? Illustration of the first law. Next, so once again, law of dominance. The three postulate characters are controlled by discrete units called factors. Second postulate, factors occur in pairs. And third, in dissimilar pair of factors, one member of the pair dominates while the other is recessive. Another name for factor is allele. Mendel gave the name factor, now it is called allele. So, this is the illustration of it, and see here you are explaining. See, this offspring has two dissimilar alleles, not similar alleles, dissimilar alleles, one capital T and the other small t. And what happens is capital T expresses because it is dominant and small t is suppressed. So, what will be the phenotype? Physical observation, we will see that these offsprings are all tall. Now, I am going to the next law. Again, it is taken from your text, wordings exactly from your text. The next law is law of segregation of gametes. Though the parents contain two alleles, during gamete formation, the alleles of a pair segregate from each other such that the gamete receives only one of the allele. So, once again, how the law is stated, though the parents contains two dissimilar alleles, say two dissimilar alleles. For example, if I continue with the same example, see, look here, I am continuing with this example. We have the parent or the F1 generation with two dissimilar alleles, capital T, small t. What will happen? At the time of gamete formation, what happens? The alleles separate in such a way that the gametes receive only one of the allele. So, to illustrate this law, I have to give the same example of the monohybrid cross. Okay. So, continuation is from here. Now, I am continuing. We worked out the cross to explain the first law. Now, from here I am continuing. P2, how is the P2 denoted? We have to self it. Capital T, small t, cross, capital T, small t, and we know both are talls. Next, gametes. Here, we have to apply the law. So, I am stating it with another color. See, both the parents have both the alleles. What happens at the time of gamete formation? The alleles separate in such a way. See, the alleles are going to separate in such a way that the gamete receives only one of the alleles. So, this gamete receives only one capital T and the other gamete receives only one small t. Such a condition, see the third condition I am giving, that the gamete receives both the alleles, does not occur. Such a condition will never occur. So, once again, at the time of gamete formation or during gamete formation, what happens? The alleles separate from one another such that the gametes receive only one of the allele, never both. Okay, such a gamete with two alleles does not happen. Okay, so I am continuing this here also. I have to proceed to F2 generation. How is F2 generation? Cross this with this. this T with this, then small t with this and this. So, we have capital T, capital T, capital T, small t, capital T, small t, small t, small t. Now, here you apply the law of dominance. This is definitely tall, no doubt about that. Now, what is this? This is also tall. We have learnt according to the law of dominance, when two dissimilar alleles occur, one expresses, the other suppresses. This is also tall using the law of dominance, and this is dwarf. Both the alleles are small t or recessive, so that is dwarf. So, we get the ratio 3:1. 3:1 means 3 tall is to 1 dwarf. So, this is a monohybrid ratio. So, once again, to explain what does the law states, though the parents contain two dissimilar alleles, at the time of gamete formation, what happens? The alleles separate from one another and each gamete receives only one of the allele, never both. So, that is explained at this step. So, you have to use the same monohybrid cross from P1 gametes F1 reach P2, and here P2 gametes, when it is formed, you are explaining the law of segregation of gametes. See, separate in such a way that the gamete receives only one of the allele. And then you cannot leave it at that, continue F2 generation, and you have the F2 generation comprising 3 tall and 1 dwarf. This ratio is also known as the monohybrid ratio. So, we can see the first law and the second law is got from the monohybrid cross. Okay. So, Mendel also proposed the first law and second law from his monohybrid cross, that is a cross where the parents were differing in one character. Okay. Now, we are going to the third law. Third law is going to be derived from a dihybrid cross. So, now I am done with monohybrid cross. I hope you have understood how to work out a monohybrid cross. You know the monohybrid ratio 3:1, and the two laws that were derived from a monohybrid cross was the first law, law of dominance, and second law, law of segregation of gametes. Now, moving on to the third law, law of independent assortment. Third law, law of independent assortment. When two pairs of traits are combined in a hybrid, I am reiterating, when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. Learn it as it is. Once again, when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. This can be well understood with an illustration, and that to which type of illustration? A dihybrid cross, where the parents are differing in two characters. Monohybrid cross is a cross where the parents are differing in one character. Now, I am going for a dihybrid cross. So, consider the cross between tall parents with violet flowers crossed with dwarf parents with white flowers. So, always when you do regular genetic problems, homozygous versus homozygous. So, you need not ever think of the other option of a heterozygous condition here. Only in genetic problems, when the question paper is asked to solve, when they ask to solve genetic problems, there you need to think of these two options, homozygous or heterozygous. But when you are doing a regular genetic problem, you have to go for homozygous condition. So, tall violet versus dwarf white. Clear? That is P1. Next, gametes. Now, before that, this is only the phenotype indicated. So, what will be the genotype? Capital T, capital P, capital B, capital B, and here small t, small t, small b, small b. I told you in a regular problem, we will go for homozygous condition. Now, gametes. Here, the gamete will have one representation of T, capital T, and one representation of B. So, capital B. Here, again, one representation of small t and one representation of small b. Then, F1 generation. This will cross with this, you get, bring the T's together, capital T, small t, capital V, small v. Okay. Now, can you guess the phenotype? Genotype is given, the alphabets are given, or genetic constitution is given. Give the physical appearance, look at this, among these two T's, which is dominant, tall, among these B's, which is dominant, violet. So, definitely this is tall plants with violet flowers. Okay, so I gave the genotype and the phenotype. Now, we are going to the F2 generation. So, to go to the F2 generation, definitely we have to find out the P2. And I told you P2 parents will be selfing of this. You have to self the F1 generation. So, I am selfing it. Gametes. Now gametes you can understand and write, but be more practical during the exam, no time to waste. So always remember, you have when a heterozygote under dihybrid cross comes, you have to go for 4 gametes. So put 4 circles, and one alphabet you double and write, and the other alphabet you alternate. So once again, be very fast and practical during exams. Need not waste time thinking and understanding and putting the gametes. Remember, a dihybrid P2 parent, the gametes, there will be 4 types of gametes, and to distribute the alphabets, remember, first alphabet you give it in double, make it double, and the second alphabet you make it in alternating form. So I am going with the alphabet T. I am going to double here. Double is, I am giving another color so that this will stand out. Capital T, capital T. So the T's I have given double. Capital T. Again I am repeating capital T, but I am repeating T, but writing small t, small t. So I filled the T's. How did I fill the T? Capital T. That one small t, small t. Now I am going to fill the V. V, how we are going to do? Alternate. Capital V, small v. Capital V, small v. This is the way to fill the gametes rather than thinking and bringing. Okay. So now let's be smart and do the next. Next one is also same. 4 gametes. Now how you are putting the alphabets? Capital T, small t, small t. Now about the V's, alternate. Capital V, small v, capital V, small v. So now we have 4 gametes of this parent and 4 gametes of the other parent. Now we have to make crosses. Okay. So now we are going to make cross. This will cross with these 4. So 1 from here, then to this, then to this, then to this. Then from the second one, I am using another color. From the second one, this to this, this to this, this. Okay. Now from the third one, I am using blue. Third gamete to all 4. Then from the fourth gamete, I am using white. Now look at the state. This is the way to cross, but very messy, very untidy. You cannot even identify. So it creates a lot of problems. So there's another way to represent it to avoid confusion. An easy method is the Punnett square. Punnett square is a tabular form, okay, or graphical representation. Now tabular form, you have to draw a table. Okay, I drew a square, and how many rows and how many columns? The one parent has 4 gametes, the second parent has 4 gametes. So one row indicates one parent, the other row the column indicates the other parent. So how many rows and how many columns? 4 gametes, no. So is it 4 rows and 4 columns? No. 5 rows and 5 columns. So 5 rows. See, 5 rows. 1, 2, 3, 4, 5. 5 rows. And how many columns? 5 columns. One. So look at the columns 1, 2, 3, 4, 5. So always remember, exam time also don't waste your time. Immediately go for 5 rows and 5 columns. Okay. Now you are going to fill the gametes. Remember, the first cell you just divided diagonally. Okay. We are not putting anything in that cell. So just strike that cell. Now we are filling the gametes of the one parent here and the gametes of the other parent here. Okay. So let's start. As I told you, how was the gametes, the alphabets distributed? I am going to work out the same. Double, double, and the other one alternate. Capital V, small v, capital V, small v. Here also I am going to apply capital T, small t, small t, and here alternate. Capital V, small v, capital V, small v. Now fill every cell. Okay, I am doing the filling. capital T, capital T, capital V, capital V, capital T, capital T, capital V, small v, capital T, small t, capital V, capital V, capital T, small t, capital V, small v, capital T, capital T, capital V, small v, capital T, small v, small v, capital T, small t, capital V, small v, capital T, small t, small v, small v, capital T, small t, capital V, capital V, capital T, small t, capital V, small v, small t, small t, capital V, capital V, small t, capital V, small v, capital T, small t, capital V, small v, capital T, small t, small v, small v, small t, capital V, small v, and last cell small t, small v, small v. Now, this 2-3, after doing 2-3 problems, you will be an expert of filling these. But remember always 5 rows, 5 columns, and distribute the gametes of one parent in the row and distribute the other gametes in the column and fill the cells. Now, when you notice and when you count the number of individuals under the following categories. For example, what were the initial parents? I told you tall violet. How did we start? Tall violet was crossed with the initial parents. Tall violet was crossed with dwarf white. Okay. So remember, 9 will be of the dominant character. So 9 of them will be tall violet. And the smallest number 1 will be the dwarf white, the recessive trait. So once again, the ratio anyway, it is, if you analyze or you verify also, the ratio is, there are 16 offsprings. Of the 16 offspring, it comes in the ratio 9:3:3:1. This ratio is called a Dihybrid ratio. Of these 16 plants, 9 will be the dominant, that initial parents tall violet, and that 1 will be of the recessive trait dwarf white. Now, how to fill the remaining 2? They are the recombinants. Now, recombinants is this. See, I am taking this tall with white. See, I am crisscrossing it. Should be tall, white. Then what about this 3? These 3? This will be dwarf with violet interchange. So this is also a recombination. Dwarf white. I hope you have understood. So when you analyze the parents or when you analyze the offsprings, you can see 4 types of offsprings are formed. In the F2 generation, how many types of offsprings are formed? 4 types of offspring. They are tall violet, tall white, dwarf white, and dwarf violet and dwarf white. Clear? So there are 4 types of offsprings formed, and of these 4 offsprings, 9 will be the parental combination and 1 also parental combination, whereas the 3s, the middle ones, are recombinants. These are recombinants. So once again, the offsprings you get as a result of dihybrid cross, the F2 generation, comprise 4 types of offsprings, namely tall violet like the dominant initial parent, dwarf white like the dominant initial parent, but 1. That one will be maximum, this will be minimum. And there are 3 other recombinants. What you have to do is you take tall white and dwarf violet. Okay, you get the recombinants. Okay. Now I am applying the law of independent assortment. Now looking at these offsprings, I am going to explain the law of independent assortment. Okay. So here I am highlighting tall. Tall is independent and can go with violet or white. So what does the law states? That the pairs of characters are independent and can assort independently. So tall is independent and can assort with violet and white. Similarly, dwarf is independent and can assort with violet or white. I hope you have understood this. It's not that tall will only go with violet and dwarf will only go with white. The two recombinants does not exist, it is not that. Just the opposite of this we are going to learn as a last pattern of inheritance called linkage. Tall will go only with violet and dwarf will only go with white. They cannot mix and match. They are not independent. Tall is dependent, it will go only with violet, and dwarf is dependent and only can go with white. That is this case. But now what we are talking is the case of independent. They are free. Tall can go with violet or white. Similarly, dwarf can go with violet or white. This is the law of independent assortment. So, explaining the law only from the observation of F2 generation of a dihybrid cross. So, you have to work the entire cross. You have to work the F1 generation, F2 generation, get the ratio 9:3:3:1 with the help of a Punnett square, and bring these 4 offsprings. And with the offsprings only you are going to explain. That is, you see, this is the initial parent tall violet, and here we have the dwarf white. These are parental form combinations, but the middle two are indicating recombinants. How did we get the recombinants? Because tall is independent and can assort with violet and can also assort with white. Similarly, dwarf is independent and can assort with violet and white. Unlike this situation where tall, instead of independent, I am using dependent, it goes only with violet, and dwarf is dependent and can go only with white. So, then in this case, no recombinants. Recombinants are not formed. This we are going to learn in the last pattern of inheritance, which is deviating from Mendel's pattern. So, once again reiterating the law with the illustration. So, once again defining the law of independent assortment. When two pairs of traits are combined in a hybrid, so what cross it is? A dihybrid cross. The segregation of one pair of character is independent of the other pair of characters. The illustration is only with the help of a dihybrid cross. A dihybrid cross, you can take any dihybrid cross. You have taken a cross between between two parents differing in 2 characters. The characters are height and flower color. One parent is tall violet, the other parent is dwarf white, and because the regular genetic problem, I am going for homozygous tall violet. Gametes are formed, F1 generation, then I am going to P2 by selfing, then gametes. I told you the technique of forming the gametes of dihybrid F2 generation. How would the gametes? 4 gametes, and then how you will assign the alphabets? Double and alternate the other alphabets, and then you have to I showed you how it will become messy when we draw the line diagram. So, to avoid that confusion, you have to go for a Punnett square, a graphical representation where you have 4 rows, 4 columns, 5 rows, 5 columns. The first row indicating the gametes of one parent, and the first column indicating the gametes of the other parent. See, this is indicating the gamete of one parent, this is indicating the gamete of the other parent, and then fill in the cells. When you fill in the cells and complete it, you will find that the ratio you get is 9:3:3:1. 9, remember, it will be the dominant parent. Which was the dominant parent which we took? Tall violet. And 1, which is minimum number, will be the recessive parent, that is a dwarf white. And we will get recombinations or new combination 3:3. The new combination will be tall white, dwarf violet. Okay, they are the recombination. So, how did we get the recombination? The answer is the law. What is that? Tall is independent and can assort or can go with violet and white. Similarly, dwarf is independent and can go with violet and white. They are independent, not dependent. I hope you have understood the 3 laws put forward by Mendel. Thank you.